name's Tara Espinosa, and I will be presenting on Query fever, also known as Q fever. So, the infectious agent behind Q fever is Coxiella burnetii. And below you'll see the kingdom, phylum, class, order, family, genus, and species, and then a picture of Coxiella burnetii. So, what are some characteristics of Coxiella burnetii? It's a small gram-negative bacteria. It's also an obligate intracellular bacteria, which means that it replicates and lives inside the host cells. It's pleomorphic, which means it alters its size and shape in response to environmental conditions, and is a coccobacillus. The three different forms are small cell variant, which is highly contagious, large cell variant, which is usually what you find in cell cultures, and then spore-like particles, which are infectious and robust to the environment. And you can see all three pictures of the small cell, large cell, and spore-like. So a little about the pathogenesis. So it is phagocytized into the host cell, so the small cell variant interacts with the cell membrane and forms a vacuole inside. And the small cell variants are metabolically inactive, and acid activation leads them to form the large cell variants, which are metabolically active, and they correspond to the intracellular form of the bacteria. So after the small cell variant help lead to the formation of the large cell, they both divide by binary fission, and these spore-like particles are formed due to the sporogenic differentiation in the large cell variants. And then, so these spore-like particles will undergo development to the small cell variant. So when the cell undergoes lysis and exocytosis, these small cell variants go out, leave the cell, and then can go infect, interact with more cells, and so on and so forth. So a little history about Q fever. So it was first recognized by Edward Holbrook Derrick, who was the director of the lab of microbiology and pathology of the Queensland Health Department at Brisbane, Australia. And he was sent to go look at around 20 of 80 infected slaughterhouse workers, and he described the disease as a febrile illness. So he tried to isolate it, and he couldn't, so he sent samples to Burnett and Freeman, who were in Australia, and they started working on it. Since Derrick didn't know the cause, he named it query fever, because he had queries about it. So Burnett and Freeman then started working on it, and they were able to reproduce the disease in mice and monkeys, and they believed that it had that it was probably in the genus of Rickettsia, so they named it Rickettsia burnetii. And then, while at this time, not knowing what was happening in Australia, Cox and Davis in Montana were working with it, which they isolated from ticks, and they named it the 9-mile agent. So while they were working in the lab with the 9-mile agent, a lab assistant of theirs got infected, and that's how they were able to link the 9-mile agent and the Australian Q fever together and realized that it was the same thing. So then a man named Phillips came in and said that it didn't really fit into the Rickettsia genus because of its genome. So they decided, and it had a lot of distinct differences, decided to put it in a new genus, which they named Coxiella burnetii after the two groups that studied it. So a little bit about the transmission. It is a zoonotic disease, which means it is um, um, transferred from animals to humans, excuse me. So with animals, they get it through direct contact, inhalation, ingestion, aerosols, um, and it's usually found in their vaginal secretions, semen, feces, urine, milk, and placenta. Now, it can be transmitted, CBRNET can be transmitted by ticks, but it's rare. Humans are usually infected via aerosols, due to the infectious airborne particles being able to travel up to 10 miles. Um, so they usually get it that way, but they can get it from ingesting unpasteurized milk um, from the infected animals. Person-to-person transmission is really rare, and so are tick bites, but they do happen sometimes. CBRNET is also can survive for prolonged periods of time in the environment, such as 7 to 10 months in wool and 120 days in dust. Some signs and symptoms, most people that have Q fever are asymptomatic. There are two types, acute and chronic. Acute is more likely, and it usually has a lot of flu-like symptoms, so fevers, chills, fatigue. You can develop pneumonia with acute Q fever. The chronic Q fever is much more rare, and the incubation period is months to years. And with the chronic Q fever, you can develop endocarditis, which is a serious health problem. And pregnant women can have reproductive losses such as miscarriages, spontaneous abortions, premature delivery, and with animals, the loss of pregnancies is usually the hallmark sign. So diagnosing Q fever, it's very difficult due to how rare it is, and it presents with a lot of flu-like symptoms. But you can use serology and PCR, which is polymerase chain reaction, to diagnose them. It's very complex, but it can be used. PCR is usually used for the acute cases and is best to be used in the first 2 weeks of infection. And with the serology, you can use ELISAs and component factors. It's very complex, but with the acute Q fever, it's usually diagnosed due to the high number of antibodies to phase 2 antigens, and with the chronic, it has high levels of IgG antibodies to phase 1 antigens combined with constant or declining titers to phase 2. So treatment. Most people don't seek treatment for the acute Q fever. That's why antibiotics are not required, but they can reduce the severity of the symptoms and reduce the course of the disease. So usually tetracyclines are used, most often 2 weeks of doxycycline, and if for pregnant women, it's usually trimethoprim and sulfamethoxazole. With chronic Q fever, it's a little more difficult to treat, so it's usually a combination of antibiotics, which involve tetracyclines and hydroxychloroquines, and used for a prolonged amount of time, usually around 18 to 24 months. And if the patient has developed endocarditis, usually surgery will be needed to repair those damaged heart valves. So risk factors. The risk factors for usually the animals that are likely to be reservoirs are the sheep, goats, and cattle. For humans, there's occupational hazards, which would be like for farmers, slaughterhouse workers, lab personnel, and dairy workers, because they usually come in contact with CBRNET due to having so much prolonged time with sheep, goats, and cattle. Location is also a big deal. Um, like I said before, the infectious airborne particles can travel up to 10 miles, so living closer to farms puts you more of at risk than living farther away from the sheep, goats, and cattle. Sex, it really is not linked to males or females. It's just males usually have those occupations, so the data shows that it's more males, but there's no direct link between why, if it's more susceptible to males or females. And immunocompromised individuals are always at risk just because um, of their weakened immune system, and patients with heart problems could develop endocarditis too. Prevalence, you can see that it's not very prevalent. Um, there's not a lot of cases. 180 is the most in a year, and you can see that the chronic Q fever rarely happens as opposed to the acute. Prevention, the Q fever vaccines are not available in the US, so there's no vaccine for it here. And then don't consume unpasteurized milk, because then if the animal had CBRNET, then there's no you will probably get Q fever. And then lastly, there is a vaccine. It's available in Australia. It's usually for vets and people that work with and will have work with sheep, goats, and cattle, and will have more contact with their placenta, urine, semen, and be more likely to get the disease. Here are my sources. Thank y'all for listening, and if you have any questions, you can ask me on Monday. Thank you.